Good afternoon, students. I just wanted to show you how to complete the Gizmos Meiosis Lab. In order to get to this lab, you're going to go to your single sign-in in Clever, log in as a student, and then click Gizmos. If you don't have Gizmos as one of your apps up here, or your favorite apps, you'll just have to go and scroll down to all the district resources and find it and add it. Gizmos is that blue G. When you actually log in, all of my classes are here, and this assignment, the Meiosis virtual lab, has already been assigned to you. So within your exploration sheet that is in Canvas, it will tell you step by step which assignments that you're going to complete. So you're going to do activity A, and you're going to do activity B. So let's go ahead and launch our Gizmo. So in activity A, you are going to be exploring the steps of meiosis. At the very top, it says, get the Gizmo ready. So make sure the steps tab is selected. And also, if necessary, make sure that you choose the male cell. And then your next step says to click on the DNA to copy it to proceed to prophase 1. So you're going to click your DNA. Now right here to the left is going to be your steps of meiosis, starting with interphase and ending with telophase 2. You also have an allele map. You are able to actually look at the chromosomes and see what dominant and recessive alleles are going to be on that chromosome. It's not necessary for this lab for you to do so, but if you just wanted to be curious, you can drag your homologous chromosomes here and actually view that. Right here at the bottom, it actually gives you a description of actually what's happening in the lab. And of course, here's your cell. Right here at the top, it actually really tells you at the top right-hand corner exactly what you need to do in order to move forward in this, along with your worksheet. So the question that we're going to answer with this is, how does meiosis create 4 daughter cells from 1 parent cell? So number 1 says to observe prophase 1. Click on the nucleus to break it down, then click on the DNA to condense it into chromosomes. So as we can see here, we need to condense our DNA. And it turns into chromosomes. Now, the typical human cell, we already know that there will be 46. This is meiosis, so there would typically be 23 of these. But for the sake of this lab, it would be 4. So after you click on the nucleus to break it down, then click on the DNA to condense it into chromosomes, and we have 4 here. And then drag the centrosomes, here they are here, to the top and bottom of the cell. Now A asks you how many chromosomes does the cell have? It has 4. Each chromosome consists of a pair of sister chromatids. And on the right, draw 2 lines connecting the pairs of homologous chromosomes and match them. So in the Gizmo, drag the homologous chromosomes together. Remember, homologous chromosomes are the same size and the same shape. So we're going to match them together. After this happens, you can click continue. Now, if you wish, you can click on the chromatid, and these are our sister chromatids held together by a centromere right here in the middle to create crossover. Remember, crossing over happens in prophase 1, and this is what gives us our genetic variability. So if you click, your chromosomes crossing over will happen here. You can let it cross over once, twice, it doesn't really matter. And you can click continue when you are done. Now next we're going to observe metaphase 1. So right here at the metaphase plate, we also call this the equatorial plate of the cell as well. We need to drag the homologous pairs of chromosomes to the metaphase plate right here in the middle, because in metaphase, all of our chromosomes line up in the middle. Now, right here on the allele map, if you want, you can drag a chromosome here to view it, to see what type of alleles ends up on the chromosome. And on this one, we have more dominant alleles than we do recessive. When we talk about genetics next week, these will make sense. If you want to observe this one, you have dominant alleles here, and you have some recessive alleles. All right, so in observe, it says to drag the groups of homologous chromosomes to the metaphase plate. We've already done that. Now we need to click the centrosomes to pull the chromosomes apart. What we're going to do is we're going to take our spindle fibers and we're going to attach them here. Remember that the spindle fibers have to attach to the centromeres in order to pull these chromosomes apart during anaphase. Now, pay close attention here. Anaphase 1 and anaphase 2 are very different. And then when we click our centromeres, they pull our chromosomes apart. But unlike mitosis, it's very, very different. Unlike mitosis, it pulls the sister chromatids apart. And so you have the single sister chromatid on either side of the poles. But in meiosis, in anaphase 1, this is a little bit different. So it says that how do the chromosomes separate in anaphase 1? The pairs of homologous chromosomes are pulled apart and each intact chromosome is pulled to the opposite side of the cell. So for number 3, an image of the anaphase step in mitosis is shown to the right. How does anaphase 1 in meiosis differ from anaphase in mitosis? So in mitosis, typically your sister chromatids are going to be pulled apart, but sister chromatids stay together in anaphase 1 of meiosis. Typically, these would be separated, this sister chromatid from this one, and they will be on opposite sides. But as you can see in anaphase 1 of meiosis, they stay intact, they stay together. At the end of anaphase 1, how many chromosomes are on each side? For that answer, you should put 2. So now it says to click on each group of chromosomes to reform the nucleus and unfold the DNA. So now we're in telophase 1. And as we can see, we have 2 new nuclei. Now right here we have a contractile ring. So during cytokinesis, a ring of proteins called the contractile ring forms around the equator of the cell. And so this ring shrinks, pinching the membrane inward and creating a cleavage furrow. And this results in the formation of 2 haploid, keyword haploid daughter cells. So what you want to do is you want to drag this and pull it to separate the chromosomes. Now remember, we're still not done. We still have about the same genetic information. The whole point of meiosis is to end up with 4 sperm cells that are all genetically different. So we have to go through this process one more time. We'll start with prophase 2. I'll click on the nucleus of each cell to break it down and to condense the nuclei into chromosomes. Then you're going to drag your centrosomes to the left and the right of your cell. Then you're going to drag your chromosomes to the middle, going through metaphase now. In order for us to pull these apart for anaphase, the spindle fibers have to attach to the centromeres. Remember, these guys only show up during cell division. Now click on each centrosome to shorten the spindle fibers and then to drag the chromatids to opposite poles. This looks more like anaphase in mitosis, doesn't it? Sister chromatids are separated. If you want to look at the alleles now, you can compare them. And you can see that all this genetic information that was in the original sperm cell has been reshuffled. Now click on each group of the chromosomes to reform the nucleus. And then finally, with telophase 2, we're going to make our cells. Now, observe that you have 4 haploid cells. After cytokinesis, you have 4 haploid sperm cells. We call these spermatids, and they're all the same size. They're all the exact same size. They're not identical. They're all genetically different, but they're the identical size. But in the next activity, you're going to figure out that the way that female and male gametes divide are a little bit different. So let's go ahead and get ready for activity B. For activity B, in order to get ready for this gizmo, make sure that the steps tab is selected and then just click reset. Now we're going to choose the female. For number 1 on activity B, you proceed through meiosis until you reach the end of anaphase 1. So we'll stop right here. So we kind of already know how to do this process. Click on the DNA, and it will condense to form our chromosomes. You drag your centrosomes to the opposite poles. Then you match up your homologous chromosomes. Click on them for crossover, because during this time when they match up, these sister chromatids will actually kind of hook up with the other homologous chromosome, and they will actually kind of swap genetic information. This always happens in our sperm and egg cells. We'll continue, and then we are going to line up our homologous chromosomes on that plate. Pull our spindle fibers out from the centrosome to attach the centromere so that they can pull them apart for anaphase. So we've pretty much done all of this. Up to this point, did you notice any differences between the development of the male and the female gametes? You should put no for that. Explain that meiosis has so far proceeded in the same way as the male cells. Now for number 2, you're going to work through telophase 1 and cytokinesis 1. Just click on our centrosomes and click on each chromosomes to reform our nuclei. Then we have telophase 1 and now cytokinesis. Now, what I want you to do is for the compare questions, what do you notice about the size of the 2 resulting cells? This one's bigger and this one's smaller. Cells really didn't divide that evenly. There's one large one and one small one. But how does this compare to the 2 cells at the end of telophase and cytokinesis for the male cells? Remember that they were all not genetically the same, but as far as the same size, they were all the same size for the sperm cells. But the egg cells will form a little bit differently. So now for number 3, continue through meiosis until you finish telophase 2 and cytokinesis. Let's go through prophase 2. Going through this again. Drag your centrosomes to the right or the left. For metaphase, take your chromosomes, put them in the middle. Your spindle fibers need to attach to the centromeres in order to pull them apart for anaphase. And then click on each centrosome to shorten the spindle. Click on the nuclei to reform it for telophase. And now for cytokinesis 2. Now observe the ovum, which is the egg cell, and these 3 polar bodies. And here's your conclusion at the bottom. Now, do all of these cells, are all of them functioning egg cells? No, only this one is. These other 3 polar bodies will end up dying later on because they won't have enough genetic information or enough cytoplasm or organelles to even carry on. So these 3 will actually end up dying. So we have 1 big cell and 3 small cells. They really don't play a significant role and they will soon die. So after doing this, your last question is to think and discuss. Why do you think egg cells are large and sperm cells are so small? Remember their role in reproduction. And remember that this cell, once it is fertilized by a sperm cell, this actually becomes our zygote after it has been formed. So these eggs have the potential to form into an organism. So they're going to need the nutrients and supplies and the cytoplasm to do so. All the sperm needs to do is transfer the genetic information to the egg cell. So they don't really have to be that large. I hope this was helpful.